Class, let us try to understand the photorespiration. It is photorespiration. A type of respiration takes place in chloroplasts in presence of light. A type of respiration that takes place induced by light, sunlight. This respiration takes place in chloroplasts. It is different from normal respiration. You know about the normal respiration that includes glycolysis, Krebs cycle, electron transport, wherein the glucose is finally oxidized to carbon dioxide and water to give rise to ATP's energy. That is different. Here it is different. Photorespiration, also known as oxidative photosynthetic carbon cycle. Oxidative respiration is oxidative photosynthetic carbon cycle or C2 cycle, because here a C2 compound will be formed as a first stable compound, first intermediate. Photorespiration takes place during active photosynthesis in presence of light. So this is another important point to remember. Photorespiration takes place during active photosynthesis. Photosynthesis will be taking place side by side, and also photorespiration in presence of light. In presence of light only the photosynthesis takes place. Apart from photosynthesis, in addition to photosynthesis, respiration also. It is present in C3 plants, absent in C4 plants. Photorespiration present in C3 plants, absent in C4 plants. Due to photorespiration, the net yield of photosynthesis decreases. Respiration is an oxidative process. The energy-rich molecules are broken down. It is catabolic type of reaction. So therefore, when there is photorespiration, the net productivity of the plant decreases. So consequently, what happens? In C3 plants, photorespiration is present, productivity will be less. Whereas in C4 plants, the photorespiration is absent, productivity more. In C3 plants, less productive than C4 plant. Another important point, here it is not mentioned. The photorespiration reactions, all reactions of photorespiration take place in three types of cell organelles: chloroplasts, peroxisomes, mitochondria. Chloroplasts, peroxisomes, mitochondria. Three types of cell organelles, all the reactions if we include. Then under what condition, what environmental conditions the photorespiration takes place? Hot, dry conditions tend to cause more photorespiration. Hot and dry. Generally, the C4 plants grow under hot conditions. Then they grow in tropical countries where the temperature is more. Therefore, the respiration. Now, this is, you can understand why in chloroplasts the photorespiration takes place simultaneously, the Calvin cycle of reactions. This is RUBP or Rubisco, RUBP carboxylase. This RUBP carboxylase has two active sites. It is a molecule, it has two active sites. Through one active site, it can absorb the carbon dioxide. Another, through active sites, it can absorb the oxygen. It has the ability to absorb two, carbon dioxide as well as oxygen. Now, when does it absorb, assimilate carbon dioxide? High carbon dioxide oxygen ratio. In other words, when there is more carbon dioxide than the oxygen, the Calvin cycle type of reactions, the Rubisco absorbs the carbon dioxide and it will form the, what is the first product? Phosphoglyceric acid, PGA. So again, from phosphoglyceric acid, Calvin cycle, phosphoglycerate. So this Rubisco enzyme, through one active sites, it can absorb the carbon dioxide. When there is more carbon dioxide than the oxygen, simultaneously when there is a low temperature. Two conditions should be fulfilled. At the same time, it can also absorb the oxygen. It can absorb the oxygen. Under what conditions? Low carbon dioxide oxygen ratio. That means more the oxygen than the carbon dioxide. More the oxygen than the carbon dioxide. Under such conditions, it will absorb the oxygen. When it absorbs the oxygen, not the Calvin cycle but photorespiration type of reactions. When the Rubisco enzyme absorbs the oxygen, so it plays a dual role. It has the ability to absorb the carbon dioxide as well as oxygen, but which one it absorbs? It depends upon the conditions. More carbon dioxide, low temperature, carbon dioxide. More oxygen than the carbon dioxide and high temperature than oxygen, the photorespiration. In photorespiration, the products are photophosphoglycerate. Phosphoglycerate, which is a C3 compound, and phosphoglycolate C2 compound. C2, C3, C5. So thus, Rubisco enzyme plays a dual role. It has two active sites on its surface. Can absorb carbon dioxide. When it absorbs the carbon dioxide, Calvin cycle type of reactions, normal photosynthesis. But when it absorbs the oxygen, preferring the carbon dioxide, the photorespiration type of reactions. Then this has two active sites. One binds to carbon dioxide, facilitating carboxylation. Another active site binds to oxygen, facilitates oxygenation. When it absorbs the oxygen, the process is oxidation. When it absorbs the carbon dioxide, carboxylation. However, Rubisco has greater affinity to carbon dioxide than oxygen. Suppose when carbon dioxide and oxygen are available in equal amounts, it prefers to absorb the carbon dioxide. So therefore the photosynthesis, that is an advantage. The key factors that are relative, the relative concentrations of oxygen and carbon dioxide. Now let us see the reactions. The photorespiration reactions. This is chloroplast. This is peroxisome. This is mitochondria. Three cell organelles. Chloroplast, peroxisomes, mitochondria. But initial reactions take place in chloroplast. Now RUBP, ribulose biphosphate, Rubisco. Let us suppose more oxygen is there, active photosynthesis is taking place, more oxygen is released. Under such conditions, the oxygen binds to the active site of Rubisco. This is 5 carbon atom compound. This is there is no carbon here. Therefore, two products are formed: phosphoglycerate, a C3 compound containing 3 carbon atoms; phosphoglycolate, C2 compound containing 2 carbon, C3 plus C2, C5. Then this phosphoglycerate, let us keep it aside, or it may go to the Calvin cycle. Then this phosphoglycolate, a C2 compound, it migrates, it is transported from the chloroplast to peroxisomes. Chloroplast to peroxisomes. So, there glycosylate, after entering into the peroxisomes, it is converted to glycine, an amino acid. This glycine, then why the glycolate is converted to glycine here only means the enzymes necessary for the conversion of glycolate into glycine may be present here. Then two glycine molecules are converted to one carbon dioxide, one serine. 2 to 4. This is 1, this is 3. Serine is 3 carbon atom compound. Now this serine again comes back to the peroxisomes. Later it is converted to glycerate, and this glycerate may be utilized for the Calvin cycle. As you know, in Calvin cycle, the first stable compound is phosphoglyceric acid or phosphoglycerate. So thus there is a provision for regeneration of phosphoglycerate, not wasting the materials. So again, 3 cell organelles: chloroplast, peroxisomes, mitochondria. Now, when oxygen is more in concentration, Rubisco binds these two, RuBP and oxygen. The immediate products are phosphoglycerate and phosphoglycolate. Then phosphoglycolate is transported to peroxisomes. There it is converted to glycine. Glycine is transported to mitochondria. 2 glycine molecules are converted to 1 molecule of carbon dioxide and 1 molecule of serine. The serine is retransported to peroxisomes. There it is converted to glycerate. The glycerate, if necessary, it will be transported to chloroplasts, where this phosphoglycerate, original phosphoglycerate, or coming from phosphoglycerate, utilized for Calvin cycle type of reactions. These are the photophosphorylation reactions. Photorespiration.